Thank you all for coming. As you could get, Rami hinted that I'm actually working on the most important region in the brain, the ventral pallidum, which is always omitted from, the reviews with the circuitry of reward. So we've been doing a lot of work in the ventral pallidum in the last 10 years or so. And today I want to tell you about a subgroup of neurons in the ventral pallidum, the glutamatergic neurons in the ventral pallidum, and some findings, interesting findings we got about them. So first of all, the ventral pallidum, for those of you who don't know it's connected with many regions in the brain. It mainly receives inhibitory input from the nucleus accumbens. It projects to the VTA, that's the classical indirect pathway, but it's also connected with many regions inside and outside the basal ganglia. It was, until about 20 years ago, it was considered a purely GABAergic region with, that inhibits all its targets, but about 20 years ago, it was discovered that there are some glutamatergic neurons in the ventral pallidum, about 10% of the neurons that they don't seem to co-express GABA, so they're just only glutamatergic. And why are they interesting? First of all, because they are in the ventral pallidum, so they are very interesting, but when you record from the ventral pallidum and you show the animal, you give the animal a cue that predicts reward, you can see that most cells increase their firing rate, but there is a subgroup that decreases the firing rate. And in a later work, it was suggested that these neurons, again, this is the same more or less paradigm, a cue that predicts reward. You see that there is a subgroup of neurons that decrease their firing rate. And when they presented the animal with a cue that predicts an aversive stimulus, an air puff, these neurons actually increase their firing rate. And they claim in this work that these are the glutamatergic neurons. And this link between the glutamate neurons of the ventral pallidum and aversion is also corroborated by behavioral work. These are two examples. One is from Tom Nasko's lab in San Diego. So they use the real-time place preference paradigm. So channelrhodopsin is expressed in the ventral pallidum. The animal has a fiber in the ventral pallidum, and it's allowed to move freely in a box. But every time the animal enters the right side of the box, the optogenetic fiber delivers light. And if channelrhodopsin is expressed in the GABA neurons of the ventral pallidum, you see that the animal prefers to stay in that side. So this means it's a rewarding stimulus. But if the channelrhodopsin is expressed in the glutamate neurons, and the light is turned on to activate them, you see that the animals avoid this part. So activating the glutamate neurons generates avoidance. And this was replicated by Megan Creed, but specifically when activating the projections of these glutamate neurons to the lateral habenula that Rami mentioned earlier. And the hypothesis is that since we know that the lateral habenula through the RMTG inhibits dopamine release, then probably the glutamate neurons of the ventral pallidum activate the lateral habenula and they decrease dopamine release. And so we became interested in these neurons, and what I'm going to show to you is actually two papers that we published about this. First of all, trying to understand what makes these neurons aversive, or more precisely, are they connected? Are their connections biased towards aversive brain regions? And the second question is, what is the role of these neurons in cocaine exposure and abstinence? So I'll start first with the first question, and everything that you will see is the work of Lirad Levy, an MD PhD student that just is about to finish. He needs to write his thesis. Anyway, so first of all, we wanted to see if these neurons connect more strongly with aversive targets versus targets that are either rewarding or at least non-aversive. So we had VGLUT2 Cree mice, and using that we expressed channel rhodopsin in these neurons, and then Liran recorded from 6 different targets in the brain. 3 of them are targets that we know that if you activate them, they generate aversive, it's aversive. So the lateral habenula, glutamate neurons in the ventral pallidum, so synapses they make on each other, and GABA neurons in the VTA. And in contrast to these, we also recorded the inputs to the dopamine neurons of the VTA, which are rewarding, GABA neurons of the ventral pallidum, and we also recorded from medial dorsal thalamic nucleus neurons, which are not necessarily considered rewarding, but it's also a major output of the ventral pallidum, so we checked also that. And we recorded from them, and we took 3 measures. At least I'm going to show you 3 synaptic measures. One is postsynaptic, and that's the AMPA to NMDA ratio. So this measure reflects, or is claimed to reflect, the density of AMPA receptors on the postsynaptic membrane. So the higher the ratio, you see, the more AMPA receptors you have, and it's considered to reflect higher synaptic strength. So this measure correlates with synaptic strength. The other 2 measures are measures of presynaptic mechanisms. They actually are thought to reflect the probability of the release of vesicles from the presynaptic terminal. We applied 2 pulses, 2 consecutive pulses, and you can have 2 measures here, the paired pulse ratio, which is the second amplitude divided by the first one, and you can also measure the coefficient of variance of the amplitude of the first pulse. Just I'm not going to dive into the why they reflect the presynaptic side, but just intuitively, if you have a weak synapse, then every time you activate it, the probability that the synapse will be activated is smaller, okay? Because some synapses will work, some not, and so you are expected to get more variance in the synaptic current. If the synapses are strong and reliable, then every time you activate it, all of them will release the neurotransmitter, and you will have, for each stimulation, you will have the same current. So these 2 parameters are inversely correlated with synaptic strength, okay? So if the synapse is strong, you will see a lower variance. Okay, back to the data. First of all, we checked the presynaptic measures from all these targets, and you can see that we didn't see any difference between the different targets of these glutamate neurons. But if you look at the AMPA to NMDA ratio, you can see there are 2 synapses that have higher AMPA to NMDA ratio than all the rest. And these are the synapses that they make on each other, on other glutamate neurons in the ventral pallidum and in the lateral habenula. Okay, so this is our first sign that these glutamate neurons of the ventral pallidum make stronger connections on aversive targets compared to rewarding targets. Now, as I showed you in the beginning, when you activate these neurons, the glutamate neurons of the ventral pallidum, you see that it generates avoidance. Now, since these neurons, as all the other neurons in the ventral pallidum fire constantly, 5 to 10 hertz. So we thought that maybe they have like a baseline aversion effect, and they maintain some kind of an aversive baseline. And if that is true, then inhibiting them should or could be interpreted by the mouse as rewarding, okay, because we're taking away aversion. And so we checked that. We used the inhibitory dreads. We expressed them in the glutamate neurons, and we used the conditioned place preference paradigm that Rami explained about before, only here we are not pairing this one side of the box with cocaine or with the drug, but we're pairing it with the inhibition of these glutamate neurons, okay? And so we have two measures of preference. One is before the manipulation, so the first time we put them in the box, we see if they prefer one side or another, and then after after conditioning one side to the inhibition of these glutamate cells, we measure the preference again, and you can see clearly that almost in all mice, they prefer to be in the side where we inhibited the glutamate neurons. So inhibiting these neurons is in itself rewarding, okay? No effect on locomotion or anything like that. And so this first part, what we saw in this work is that the glutamate neurons seem to be connected more strongly with aversive targets, and if you inhibit them, it generates a rewarding sensation. Now, do they have a role in drug use, drug exposure? And specifically, we are interested in abstinence because that's the period where aversion comes into action. And so, sorry, we are asking two different questions. The first one is the behavioral effect. Will we see any effect on the preference of these mice to cocaine if we manipulate the glutamate neurons? And so this was, there are some experiments done before us showing, so these are two experiments. One is with cocaine, this one is with heroin. Both of them show that in a drug self-administration model, after extinction, you give the animals a cue that reminds them of the drug, and then they reinstate drug-seeking behavior. You see that both animals reinstate drug-seeking behavior if you don't do anything, but if you activate the glutamate neurons, this reinstatement of drug-seeking behavior is gone or strongly inhibited. Again, we wanted to see what happens if you inhibit these glutamate neurons, because it seems, again, if inhibiting them is rewarding, then maybe it will also affect how mice experience cocaine, or at least remember how rewarding was cocaine. And so here we used the cocaine conditioned place preference, like Rami explained. We conditioned one side to cocaine. Again, we had the inhibitory dreads in the glutamatergic neurons, and we injected the mice with CNO, the ligand of the dreads, 15 minutes or 20 minutes before the test, measured preference, and then mice went through the whole thing again, reconditioning, abstinence, and then they were injected with vehicle for control. Half of the mice got vehicle first and then CNO, so it's, the order is controlled. And again, you can see that inhibiting the glutamate neurons is not only rewarding by itself, but it increased the preference for cocaine. Okay, so now they go more robustly to the cocaine paired side. Again, no effect on locomotion. And here, because we know that the connection with the lateral habenula is probably special for these neurons, we we wanted to see if specifically inhibiting the pathway of these neurons to the lateral habenula replicates this behavioral effect. And indeed, you can see again that when you inhibit specifically the projection to the lateral habenula, again, it enhances cocaine reward or cocaine preference. We did the same thing, but inhibiting the projections to the VTA. And here we did not get, on average, a change in the preference for cocaine. But if you look closely, it looks like in every animal, we got a change of the behavior. It's just the change is not consistent between animals. So there might be some nuances that we need to understand better in order to understand the projection of these glutamate neurons to the VTA. Okay, so the last part of this story is trying to understand not how these neurons affect behavior, but how does the process of being exposed to cocaine, then abstinence, and then being tested, whether this affects the synapses that these glutamate neurons make on their target. And again, we are expressing channelrhodopsin in the ventral pallidum glutamate neurons. And because we are interested specifically in the abstinence part, so we recorded at 3 time points. One time point was right after we finished conditioning the mice. It's actually 1 day after we finished the conditioning part. Okay, so this is 1 day of abstinence. Then we recorded 14 days after 14 days of abstinence. Okay, so if we compare this time after 14 days of abstinence to this time point, we can understand how abstinence affects the synapse. And we also had another group of mice to which we put them back in the box, in the CPP box, and also injected them with cocaine before we put them in the box. And the injection, the purpose of injecting them with cocaine is to terminate abstinence. Okay, so they started here abstinence, they went through abstinence, and then we terminated abstinence and put them in the box. To control for this injection of cocaine, we also had animals receiving saline injection and then put in the box, and also another group of animals that we just put in the box without any injection. Now what I'm going to show you, I'm just showing you the highlights. It's a lot of synapses in a lot of groups. I'm showing you the highlights, and in all the graphs that you will see now, they are all 5-bar graphs. All the bars are compared to the 14 days abstinence bar. Okay, this is the prolonged abstinence bar. So you have the one day abstinence, 14 days abstinence, and then the 3 conditions of the test with cocaine injection, saline injection, and no injection. First of all, so this is the results of the AMPA to NMDA ratio. You can see this is the projection, the synapse to the lateral habenula. This is the synapse to the dopamine neurons in the VTA. So this is an aversive synapse, and this is a rewarding synapse. And you can see that abstinence caused an increase in the AMPA to NMDA ratio in the lateral habenula, but at the same time caused a decrease in the AMPA to NMDA ratio in the pathway, in the synapse to the dopamine neurons in the VTA. So abstinence kind of biased the system, and now these neurons activate, or at least we think they should activate the lateral habenula more easily than the dopamine neurons. Interestingly, when we terminated abstinence with a cocaine injection, you see that the lateral habenula synapse goes back to baseline or to pre-abstinence, while the VTA dopamine synapse did not respond to that. So it looks like this is a first hint that the synapse to the lateral habenula is really sensitive to the presence of cocaine. So this was postsynaptic mechanisms. And these are the presynaptic mechanisms. Again, these are two examples, or the most interesting data that we got here. Again, you can see in green the lateral habenula, which seems to be really strongly connected with the glutamate neurons, and also the VTA GABA neurons, which until now did not show nothing interesting, but now we found that there are presynaptic mechanisms going on in there. So we're talking about the paired pulse ratio and the coefficient of variance. I'm focusing on the coefficient of variance because results are stronger, but it looks the same as the paired pulse ratio. In both projections, you can see that abstinence decreased the coefficient of variance, also the paired pulse ratio, okay, in both of them, inversely correlated to synaptic strength, so the synapse became stronger in both these pathways. Both of them are aversive pathways, okay, to the habenula and to the GABA cells of the VTA. And injection of cocaine and terminating abstinence brought the synapse back to baseline in both of them. Now, up until this point, these two synapses look like they're doing the same thing, okay? Abstinence makes them stronger. Injection of cocaine brings them back to normal. But if you look at the two controls, so let's start with the lateral habenula. You see that animals that got saline injection instead of cocaine or no injection, this synapse did not go back to baseline. Okay, so in this synapse, it looks like these changes are driven by the actual injection of cocaine, the presence of the substance. In the VTA GABA neurons, the synapse went back to baseline even when injected with saline, and almost, it did not come out significant, but also when just putting the mouse in the box. Now the injection, the box itself and cocaine are all cues linked to receiving cocaine, okay? And so from these data, we think, and it still needs to be tested, but we think that the lateral habenula, the synapse to the lateral habenula, is linked to or is sensitive to the presence of cocaine, to the feeling of the animal at that moment, while the synapse to the VTA GABA neurons is more sensitive to the cues, to the learning, to reward learning. And so this is the summary, okay, summary of this last part. So at baseline, you see these two aversive synapses to the lateral habenula and the VTA GABA. Abstinence potentiates both these synapses, the lateral habenula both pre and postsynaptically, VTA GABA only presynaptically, and cocaine re-exposure brings them back to the pre-abstinence condition, but in the lateral habenula, it's driven by cocaine probably, and in the VTA, it's driven by the cues. Okay? So that's it for me, and this is the work again of Liran with the help of everyone in the lab, and thank you for listening, and I can take questions.